With the release of the film Contagion, an opportunity exists for the scientific community to engage in a public discussion regarding the real-world possibility of a pandemic outbreak caused by an unknown pathogen. We've interviewed 5 highly regarded virologists and microbiologists to discuss the current state of understanding and preparedness and the scientific methodologies they employ for just such an outbreak.
A couple of years ago, I had a meeting with Scott Burns and Laurie Garrett to talk about a movie that would be concerned with an outbreak of infectious disease. Soderbergh and Burns, the director and the screenwriter, had been on another film where someone had sneezed. And this was the catalyst for talking about what might happen if somebody became ill on set, and this expanded and became a major outbreak. And so we met to talk about various scenarios about how an outbreak might emerge and how it might evolve and how it could be addressed and what were some of the issues that might come up, political, economic, scientific, logistical, and so forth. A number of outbreak stories have been covered in film. One of them was called Outbreak. There were some others as well. I Am Legend. We've even got Invasion of the Body Snatchers, if you want to think about that as an infectious disease. But this is different, and this is why I became involved in this project, because it was a commitment on the part of the director and the screenwriter and the producers to make something that would be viable, would be plausible. It would describe the various aspects of the emergence of an infectious disease and would both entertain as well as educate. And I use the term education in a very broad sense. It would actually highlight some of the things that we needed to address weaknesses in our current public health system.
The Tahoe Research Initiative is a small business that is pushing diagnostics and discovery technology out internationally in order to support United States government priorities and goals in surveillance and detection of infectious disease. We've been in operation about a year and a half. Our major focus right now is that we're working in the Philippines. We have 2 partners out there, St. Luke's Medical Center in Quezon City at the Research and Biotechnology Division, and at the University of Philippines, Los Baños, where the veterinary school is. And so what we're doing is we're putting some technology in the field. Those are handheld thermocyclers, microarray technology, and of course the keystone of this project is the 454 GS Junior that we have at St. Luke's Medical Center. It's an interesting program because in the Philippines there are anti-biopiracy laws where you can't export either the microbes themselves or any of their derivatives such as proteins or nucleic acids. So if you're going to do the kinds of work we want to do, defining the local microbial biodiversity, doing sequencing, doing detection work, you have to do it in country. You can't farm it out to other parts of Asia or back to the United States. So right now what we're doing is we have the sequencer running. We have a couple of projects right now where we're working on 2 separate outbreaks. One of cholera in the island of Palawan, where we have environmental samples as well as a couple of clinical samples, and we're trying to match what we suspect is the source of the cholera with the exact strain of the bug that's coming out of the patients. So that's the kind of work that we're doing. We're trying to enable, we talk about pushing the technology to the problem and not trying to bring the problem to the technology. And so the GS Junior is what we're using right now to do the very nitty-gritty, fine detail discovery of what bugs are making people sick, where they're at in the environment, and how they compare with other strains that we know from other sequencing projects around the world.
I'm Marcio Nunes, Director of the Center for Technological Innovation, which was established 2 years ago as a new branch of the Evandro Chagas Institute, a very important Brazilian Ministry of Health institution based in the Amazon region, northern Brazil. Our institutional mission is to contribute to the development of biological science in the region under 3 basic aspects: basic science and research, eco-epidemiological and environmental studies, and public health. In this case, our main focus are the hunting of human pathogens and quick response to outbreaks. Why Brazil? So Brazil is a tropical country with one of the greatest animal and, of course, microbial biodiversity. Many important emergent and re-emergent viruses have been detected in Brazil, causing outbreaks, leading to impressive public health impact. As example, we can cite dengue viruses. In the Amazon region, many viruses and other pathogens can determine the same clinical symptoms. As example, Oropouche viruses, flaviviruses, group C viruses, and dengue viruses. Thus, the sequencing capability is important for development of specific diagnostic tools for tropical disease. In case of an outbreak situation that we call an alarming situation, our field staff is sent to the area for collecting sample. In this case, to collect a sample for patient, arthropods, and vertebrates. All samples are sent to the lab storage in liquid nitrogen in order to be processed in different ways for virus isolation, serology, and molecular biology assays. In case of molecular biology, our workflow for pathogen detection includes primary screening using mass tag PCR, a second screening using microarrays or bioships, and finally, a 454 sequencing. In 4 days, we have a great chance to discover the pathogen in the samples. Global deadly outbreaks had been occurred in the human history due to natural infections or biological terrorism activities. In fact, my personal opinion is that it's very difficult to predict when or how an outbreak will happen. Is there a possibility really to occur a global deadly outbreak? Yes, there is, but in fact, who knows? On the other hand, prevention is my answer. Knowing in advance your enemy is the key for the success. Hunting new pathogens, isolating and studying their genetic and protect material is going to be the answer when they emerge.
From the SARS epidemic, quite frankly, we weren't all that prepared. It hit us a bit of a surprise, but that was a learning experience. And since that SARS incident several years ago, we basically have backup plans to deal with the next challenge. I got a call from the Department of Microbiology, KY Yun, saying that he has 2 isolates from the recent scarlet fever epidemic. One was isolated from a deceased patient, and the other one was isolated one in intensive care. And he asked, well, if I hand you the vial personally tomorrow, how soon can you do it? And I call people back from vacation. We shut down everything that's non-essential. We purge all the bioinformatics jobs in the server in anticipation for the sequence. The bioinformatics people were asked to come in over the weekend. So KY Yun personally handed me the vial on Tuesday night. By Wednesday morning, we started making libraries. On Friday morning, it went on to 454. And by Friday night, we have raw data. The informatics crew started working on it on Saturday using Nubler. So we got the first draft off on Sunday afternoon. And by Monday morning, there was enough known about the pathogen to start designing PCR-specific probes for detection. And then meanwhile, of course, the draft was in pretty good shape. We were able to reduce it down to 47 contig using Nubler. And it looks very similar. This is purely de novo. And then by looking at that sequence, we found it's very related to another species of bacteria, another strain in a database. And using reference-assisted assembly, we reduced 47 contig down to 2. So we essentially accomplished our mission. From the time we handed the DNA sample, we went on this 454 2 days later, and a day and a half, we have the first reference draft. So it actually worked. I mean, our timeline was aggressive, and we actually did it.
I'm the director of the Center for Infection Immunity here at Columbia University at the Mailman School of Public Health. We receive some 40,000 samples a year. Outbreaks are fortunately much less frequent, but we receive outbreak samples as well. And there are advantages to working up outbreak samples as opposed to one-of or surveillance samples, because you can look for consistency amongst the samples in terms of what's present. We typically use a stage strategy for microbe hunting. We start with a very inexpensive method like multiplex PCR, which we typically do with a mass spec platform, something called mass tag PCR that we use here. When that fails, we move on to other things which might be microarrays, but increasingly we use high throughput sequencing. And the reason is that high throughput sequencing is sensitive, it's definitive. So if we actually get a sequence and we can compare it to a database, we know that what we find is not just a signal, a shadow of something might be present, but it's an actual bona fide fingerprint of the agent itself. And the advantage, of course, of using high throughput sequencing technologies is that you can get a lot of information very, very quickly. So this can enable people to select the appropriate regions to make diagnostic tests, to begin thinking about proteins that they might want to express to make immunological assays, or to think about vaccines and so on. I've been working with high throughput sequencing chiefly on the 454 platform since 2004, 2005, just as the technology was getting off the ground. And in that time, we've discovered or characterized over 400 new agents. So I've been very, very pleased with what we've been able to achieve by using this platform.
This is ILRI, the International Livestock Research Institute, based in Nairobi in Kenya. My name is Steve Kemp. I work in the biotechnology theme here at ILRI. We're working on a number of projects all around livestock, livestock health and livestock genetics. My particular work is involved in two aspects of livestock health. One is understanding host pathogen interactions, how some strains of livestock tolerate disease and remain healthy and productive despite disease challenge. And the other side of that is study of the pathogens themselves and trying to understand pathogen diversity, pathogen emergence, and look for novel ways of detecting pathogens. We were funded by Google.org to work together with a number of other Kenyan institutes to develop some novel methods for detecting viruses, particularly for detecting previously unknown viruses and potentially emerging viruses. And that's a very big technical challenge because it's the classic we don't know what we don't know situation. If we know what we expect, then we can look for those sequences. But looking for something unknown is intrinsically challenging. And so one approach to that is to use a blind mass sequencing method, and that's what the 454 brings. It brings the ability to very quickly screen through thousands and tens of thousands, in fact hundreds of thousands of reads of DNA or RNA in order to look for unknown things. So we'll find a lot of host in there, we'll find a lot of bacteria in there, but hopefully we'll also find signal from things that we've not seen before, which might represent interesting new pathogens and viruses. So the next emerging infectious disease is expected to hop from livestock or from wildlife into humans. And Kenya is a good example of a country where there's intense livestock keeping, there's lots of wildlife, and there's lots of people, and all of those three living together very closely provide lots of opportunity for pathogens to hop between species. And it's that hopping between species that's likely to give rise to an emerging infectious disease, because a virus that's well adapted for one species often causes no disease in that species, but if it jumps into humans, then it can cause very severe disease. So we're using the Roche GS Flex together with some other instrumentation to provide a pipeline for detecting new sequences and, having detected them, characterizing them. And it's an approach that would simply have been impossible just 5 or 6 years ago, because without high throughput technology, without a sequencing platform that produced tens of thousands of reads of reasonable length, a few hundred base pairs at least in length, it would simply be impossible to do this kind of work. The Google-funded project is in principle looking for any virus, but in practice it's focusing on Rift Valley fever, which is a very important disease of both humans and livestock. It's a classic zoonotic disease. And with the 454 we've been able to detect novel variants of the Rift Valley fever virus. Other examples of things we're working on are ASF, African swine fever virus, which causes important disease in pigs, and quite seriously limits the productivity of pigs across the whole of Africa. And we're trying to understand the dynamics of ASF virus across Africa. It looks like there's lots of variation in the virus, and it also looks like there's lots of variation in the host pigs, which means that sometimes you get very severe disease and sometimes you get very mild disease. And using 454, we can look at host diversity, and we can look at viral diversity and try to understand the epidemiology of that disease. There's no doubt that what has changed in the last 20, 25, 30 years is that the world really has got smaller, and population densities have increased enormously. So people are now living very close to their livestock in very dense peri-urban environments. Wildlife are coming in and mixing it up in a completely novel way, and at the same time those people are capable of flying around the world within a few hours. And so those kind of contacts, which previously must have been happening but in completely isolated pockets and thus not allowing a disease to progress, the world has reached the point now where there is so much opportunity for pathogens to hop between species, and so much opportunity for infected people to move so quickly and reach new populations of vulnerable people, that there's no doubt there really is an increased risk of emerging infectious disease. So I guess in summary, at the moment we could respond to a disaster, but we couldn't prevent a disaster.
There are a couple of challenges in pathogen discovery that go beyond the idea of detecting nucleic acids specific for an agent that many people don't think about. They think, you find the nucleic acid and you're there. But to prove the linkage to disease is frequently much more complicated. So, there are different levels of certainty that you've actually found what you want to find. So if you can grow the organism and introduce it into another animal model and reproduce the disease, that is to say, fulfill Koch's postulates, that's the most compelling information that you can possibly obtain. But there are instances where you don't have an animal model, or you can't propagate the agent in a laboratory, so you can't get that far. So then you need to do other things like look at the distribution of the agent in the affected tissues. Try to develop serological assays so that you can look for evidence that there's been a seroconversion indicative of an acute infection. This is possible, obviously, with diseases like pneumonia, encephalitis, gastroenteritis. But if you're looking for causes of cancer, for example, or neurodevelopmental disorders or diabetes, and you find an agent, it may have occurred, that infection may have occurred in the remote past, and it's much more difficult to demonstrate the significance of the relationship between what you find and the disease. So the big challenge in pathogen discovery is frequently taking it from the level of finding a footprint of an agent and moving forward into proving causation.
I'm excited about the movie. It's going to be a fun movie, and I also think that it's going to raise public awareness. I don't look at it as being it's not far-fetched. It's not involving aliens crashing on the moon. It's not some technical leap that we don't know about. We have the technology. It's realistic, and again, I think a little fear is good, because I think it's going to raise the awareness of infectious disease. This is by no means infrequent event. And I think, you can worry about things like an asteroid crashing into Earth, and I know NASA's looking at that, and they should look at it. But it's a very high impact, but very, very low probability. This isn't a high probability, this is a given. And so I like the fact that there are movies, and there's a lot of talk in the policy shops right now about how we're going to approach the infectious disease problem, because it is real. It's not farcical, it's not a maybe, it is a given.
All of the people involved in the film Contagion, were cognizant of the fact that they had two objectives. One, of course, was they had to bring people into the theaters to see the movie. So the art had to be good, it had to be a compelling story, there had to be excellent character development, it had to be scenically important. And it's pulling together all those aspects, the music and the sets and the costumes and the actors who were brilliant, and the directing, which is brilliant, and so forth. But the other aspect, and this is what I focused on, was to make sure that the science was as accurate as it could be. Now, it is not a documentary, and I don't think you're going to get, hundreds of thousands of people to go and see a documentary about an outbreak. But as far as it goes in terms of the genre of outbreak films, I'm very proud of what the group has done, and I'm honored to be associated with it, because I think that by and large it's excellent science. And our hope is that, and this is not just my hope, it's the hope I think of the entire group that's been involved with this, is that we will realize that the scientists are the heroes of the piece. And we're trying to encourage kids to go into public health and into microbiology and to find ways in which they can contribute to global health.